All right, so yeah, you're not going to get coloured handouts all the time, but this one is a special occasion. And so what I've done is to just choose some examples here. I mentioned these restriction endonucleases on this slide here for cutting out sequences in a donor organism's DNA. You also have to cut using the same endonucleases where you're going to insert this gene. If you think about it, you've got to, because you're going to need matching bases. And restriction endonucleases are specific for certain base sequences. Now, that little table at the top of your sheet there, you've got 5 different restriction endonucleases listed, and it shows you the sequences that they cut at. Now, we describe those sequences in a particular way because they read the same in the opposite directions, if you know what I mean. Okay, so if you look at that top one, you're getting a cut in a sequence that goes AGCT, and if you look at the the opposite, the other strand, the complementary strand, you've got TCGA. So you've actually got sequences that read the same in both directions, if you know what I mean. What would you call that in English? A word that reads the same backwards and forwards. Begins with P. It's a palindrome. Yeah, palindrome. So we describe these as palindromic sequences. All right, these are palindromic sequences. So they, they read the same both ways, and you can see that with all the others. Now, they have rather strange names, but those names come from the bacteria that they've been extracted from. Can't tell you what the first one is. The second one, HAE3, I think it is. It's always difficult to tell with sticks. They could be I's, they could be L's, they could be 1's. That comes from Haemophilus aegyptus, that one. BAMH1 comes from Bacillus amyloliquefaciens. HIND3 comes from Haemophilus influenzae, oh, and then it's D3. And E. co, if you had to guess which bacterium? E. coli, yeah, so their names are derived from the bacteria they've been taken from. Now you get 2 types of cut. If you look at the top 2 there, they produce what are known as blunt ends, so you just get straight cuts straight between the bases. The other ones, you can see you've actually got an overlap, you've got a step. Now what that produces is like the diagram underneath that I've given you there. It produces, and they're described there as sticky ends. All right, now sticky ends are the preferred sort of cut, because if you think about it, what you're trying to do eventually is join 2 pieces of DNA together. Now it'd be like trying to join 2 pieces of paper together. If I give you 2 pieces of paper and say, right, join these together to make a long piece, are you going to try and glue them end to end? You're probably not. You could do, to get the longest possible piece of paper, you could, if you're careful, glue them end to end, and that's your blunt cut, isn't it? But if you glue them overlapping, you get a stronger join, don't you? You're making more new bonds to hold them together. So sticky ends are the preferred sort of cut. Now, these restriction enzymes then are one type of enzyme that you get in genetic engineering. Then I've listed some of the others. The reverse transcriptase we had here, converting messenger RNA into single-stranded complementary DNA. Then DNA polymerase to make that into double-stranded DNA, and then ultimately we'll need DNA ligase to actually join the pieces together. But of course we've met ligase when we did replication, haven't we, for joining the sections together. And I just threw in a few of the examples that we'd mentioned: bacteria that produce human insulin, growth hormone in milk, pest-resistant crops, herbicide-resistant crops. I missed off the golden rice there with vitamin A in it, but you can always add that on the bottom if you haven't got it anywhere else. So we now need to move on a bit. The other thing that we had looked at was PCR, the polymerase chain reaction. Did we look at the animation for that? We did. Okay. So that's, yes, we did, didn't we? Because so we can move on, because I knew we'd be a bit short of time this morning. The next thing to think about then is how you actually modify cells. All right, you've got a bucket full of these genes now. All right, you might have made it in the lab, you might have cut it out using restriction enzymes. You've used PCR to make several million copies. We've got a beaker with some genes in. Great, what are you going to do now? Well, the next problem is, you know, how do you get it into a host cell? And can you get it into all the cells that you want to? And if not, how are you going to identify the cells that have been modified against the cells that haven't? So, you know, actually modifying cells is the next sort of step in the process. The first thing that you're going to need is something to carry the new genetic material into the cell. And we talk about this in the same way as carrying diseases, and that's with vectors. All right, so you need a vector for the DNA. So a vector would carry the information. Hang on. Right. Okay, back to this. Yeah. Okay, a vector then is something that carries the genetic material in, and a very common vector comes from the little loops of the extra loops of DNA in bacterial cells. Can you remember them from when we plasmids? That's it. Yeah, bacterial plasmids are commonly used. There are other options. They can pretty much fire the genes into cells. They do actually have what they call a gene gun. And all they do is they just coat the DNA in little tiny lipid droplets. And then all you have to do is to sort of, you know, fire it at the cells. And of course the lipids will go through the membranes, won't they, and carry the DNA in. So, so you have this, yeah, gene gun. But yeah, so you know, there's something to so, yeah, it's a good topic for a presentation maybe. But you know, I don't know anything about the sort of technology that's involved there. But the bacterial plasmids are the obvious way of doing it, and the way this works is what you've got on the other side of your sheet, and I put two examples on there. The top one is the one that we always talk about, the most obvious one. And I don't want to talk about it in a lot of detail because somebody might choose to do this for their presentation. It is quite a good one to do. If you did decide to do production of human insulin, you have to talk about the structure of insulin as a protein, where the gene is, and all that sort of thing. And in fact, this isn't entirely accurate because here it shows the human insulin gene being cut out of DNA. In fact, this one is more commonly done tracking down the messenger RNA and then reverse transcriptase and all that, because you can get more of it, so I understand. But let's assume that we've got a lot of copies of the gene. It doesn't really matter how, does it? So we've got that on the left-hand side, and then to the right you've got this bacterial plasmid. And if you remember with bacteria, never mind all the stuff around the outside, you know, you have the loop of ordinary sort of DNA, just you know, the equivalent of eukaryote chromosomes, and then you have any number of, I'm not sure what sort of numbers they come in, but these little tiny loops of DNA that only carry a few genes. Some of these have quite particular uses, actually, some of the genes in these, and I'll tell you about one specific one in a minute. But if you can kill your bacteria, and you can actually extract these. All right, so these plasmids can be taken out, and if you cut them open with the same restriction enzyme that you cut your gene out with, all right, I know this doesn't quite apply with this, but if you use the same sequence to be cut, then of course your gene will have the complementary bases to be joined in. So The first step is you take your bacterial plasmid, you cut it open, and you use DNA ligase to actually stick the insulin gene into the plasmid. That's fair enough. That bit does happen. Now, the next bit's quite tricky. Plasmid put into bacterium. Put. Okay, come here, little bacterium. You know, open your mouth, in it goes. How are you going to do this? Well, this is a practical we used to be able to do, but it's a bit time consuming, so that's one of the things that's disappeared actually. But you have to incubate bacteria with modified plasmids. And we used to get this kit of bacteria, and ready modified plasmids, I think, was what we were given, because actually doing it ourselves would never have produced enough. But you have a little tube of modified plasmids, you mix it with a bacterial culture, incubate them together at a nice temperature for a certain amount of time, and hopefully some of the bacteria will take up those plasmids. But let's say we've got 10 million bacteria. We might only find that 10 bacteria take up the plasmid, the modified plasmid. So the step that's actually missed out on diagrams almost always is, how do you identify which bacteria have taken up the plasmids, the modified plasmids? So you have to have a think about that, you know, because they're not going to put their hands up, they're not going to be obvious. You can, yeah? Go on. You could use a fluorescent tag, yes. You could, when you insert your, look, this is the plasmid, and let's say here's the inserted gene, just for simplicity. How do we know if a bacterium's got that? Well, we could insert another gene alongside it that made those bacteria evident, yeah? And actually, the one, well, you could use a fluorescent tag, or what they very often do, think about bacteria. What could you modify a bacterium to be? Resistant to, well, antibiotics, yeah. Now you're thinking to yourself, but why would you do that? Aren't you? The thing is, what we tend to misunderstand is that antibiotics are not just medicines for human diseases. Antibiotics are substances that kill bacteria. There are no end of antibiotics which will kill bacteria that are no use as medicines whatsoever. So we can use one of those. We can actually create a gene that gives resistance to an antibiotic that isn't medically used. And the one that's very often used is an antibiotic called kanamycin. But you know, there is this sort of misunderstanding that, oh, we're creating antibiotic-resistant bacteria, and you know, that is a bit of a worry. But if alongside this insulin gene that we've got here, we put in an antibiotic resistance gene, all right, so if, let's say that one is the insulin. Then, of course, all you've got to do is to grow these in culture that contains the antibiotic, yeah? And the ones that are modified will survive and grow and produce insulin, and the ones that are not modified will die, and they won't use up all your resources, you know, the food that you're giving. But because you might, I have no idea what the likely success rate is, but you know, if you were to get 10 bacteria modified out of 10 million, well, you know, you've got 9,999,990 that are, you know, using up your resources. So you only want the 10 modified ones. You know that they will multiply very quickly when you culture them anyway, but you need to be able to, you know, to identify them and extract them. So tend to grow them on agar plates with that antibiotic in, and that then enables you to culture the ones that survive. So the ones that survive the antibiotic are the ones that have been modified. We do use fluorescence labeling for other sort of characteristics. I think I mentioned that in animals, didn't I? Possibly. It might have been the other group. But they've done things like modifying pigs. They're trying to get pigs to produce what are essentially human organs, but this is a bit difficult. And to work out which ones are modified, they've put in a fluorescing gene, and you get these pigs whose noses glow yellow in the dark and that sort of thing, you know, or under UV light or stuff like that. So you can use, you know, different types of labeling, but antibiotic resistance is it's a useful one in bacteria. So we've incubated the bacteria with the plasmids. Some bacteria have taken up the plasmids. We grow them on plates to show antibiotic resistance is, you know, as an indicator of the modification, and then stick them in big tanks and they'll churn out insulin for us. Very convenient. Now, the other one that I've got on that sheet is a slightly different approach, and it's quite difficult to get the DNA into eukaryote cells, particularly plant cells. You know, they've got a wall around the outside and this sort of thing, so you have to take a slightly sneaky route. And the bacterium that's being used in that particular example, Agrobacterium tumefaciens. Now, tumefaciens, what does that suggest? Tumour, yeah. And that's what this bacterium can do. This bacterium can actually create tumours in plants. And it has, if you look, if you look down that left-hand side, you've got a thing that's labelled the T1 plasmid. Now, the T1 plasmid is, usefully, for us, for GM purposes anyway, what that bacterium injects into plant cells to cause tumours. So you've immediately got a mechanism for delivering DNA into plant cells. All right, so it's you know, it's quite a sort of convenient process there. Where they've labelled tDNA, I think T there stands for transfer, or you know, generally just that bit it's coming from another organism. So if you modify the T1 plasmid, the tumour-causing plasmid, and the bacteria are allowed to, well, you get the modified plasmids back into bacteria as before. You get the bacteria to infect plant cells. They will deliver these genes that we want for things like, on the board there we've got things like herbicide resistance, pest resistance, that sort of thing. And so those are delivered in, you know, in a slightly different way by an organism, still a bacterium, that happens to infect plant cells by injecting plasmids. So it's a, you know, it's a very convenient route, really, for you know, for getting the gene in. Okay, these genes that we use for identifying the modified cells are called marker genes. All right, so that's one example there, and we will stop at that point, and I'm going to give you a crossword to play with. Okay, so this idea of DNA probes or gene probes, the two things mean the same thing. A DNA probe and a gene probe are exactly the same thing, and they are nothing that's too difficult to understand. But the reason we're doing this is because when I rewrote this unit last year when we were revalidated, I decided it would be sensible to refer to the new A-level biology specification to get up-to-date content. You know, you're going to be going to university with people who've done A-level biology, so it's quite useful to have studied some of the same things. It's also useful for me to be able to assess using A-level biology questions, because then I know you're being assessed at exactly the right standard. So we had this bit turn up in the A-level spec on DNA probes, and I thought, actually, when it comes to it, it's not terribly complicated. Some of the techniques are, and I'll just refer you to do a bit of extra reading on those, particularly if they come up in your presentation, which they are likely to come up in somebody's. So we're not doing it in huge detail, but there is potentially quite a lot of ethical sort of debate and arguments to consider on the use of DNA probes. Now, they have a lot of uses, but the principle is quite straightforward. And it just relies on the very basic base pairing that you get between complementary bases on the two strands of DNA. Now, you know, because we know that A always pairs with T and C always pairs with G, if you want to find a specific sequence in an entire genome, all right, bearing in mind, you know, how big an entire genome is, which I've forgotten exactly what the number of base pairs there was. It was a number that was in my head at one point. But, you know, within the entire genome, if you want to find a base sequence, then it's not that difficult to do, given that we know what the complementary bases will be. And that's all that a gene probe is. If you've got a sequence of bases that form a gene, let's say this is our gene, and it goes, I don't know, I'm just going to do a repeating bit here and there. Doesn't really matter, actually, thinking about it. All right, if that's a very short gene, if we want to identify that in somebody because it's a gene for a particular genetic condition, or it helps to identify a particular type of cell, for example, cancer cells tend to express proteins differently because they've got particular gene mutations. If you want to be able to identify specific cells carrying specific genes, you can use a gene probe to do that. Now, despite the fact that the DNA is a double-stranded structure, you know, with the two strands held together, if you introduce a bit of single-stranded DNA, it will actually bind to any bit of sequence which is complementary. And if you quite simply label that piece of DNA, that, you know, this little short piece, with, well, you've got two options really. Fluorescent labels are one option, and the other one would be radioactivity. And if you label your little marker, if you like, mix it in with the DNA, actually you can separate the strands quite easily. All you've got to do is heat them up anyway, haven't you? If you think back to the polymerase chain reaction, if you've got double-stranded DNA, if you just heat it up, the two strands will separate. If you then mix in your little marker, then it can find, it doesn't have to find the entire gene. The entire gene might be hundreds or even thousands of bases long. But if you've got a very specific little target, let's say that this gene is quite unusual in that it has a bit that goes, oh, CGG, GCC, this little bit here. If you mix in with this a gene probe that has the complementary bases on it, just that, fluorescently labelled red, let's say, because that's what I've done it in, you can find it. All right, it is that straightforward. All we're doing is finding little bits of complementary code, making short pieces of single-stranded DNA, labeling them either radioactively or fluorescently, and then bringing them together. Okay, now that thing there then is our labeled DNA probe or gene probe. This might be our whole gene here. And the process of bringing them together is known as hybridization. Because obviously, you know, a hybrid is a mix. You know, if we talk about a hybrid organism, it's, you know, having mixed together two genotypes, perhaps you could have two pedigree animals. If they interbreed, you get a hybrid. And so bringing together DNA from different sources and getting them to join together is known as hybridization. And the idea is that you can actually identify, you know, some very, very specific little target sequences very, very easily. Now, in terms of the techniques that are used for this, there are some quite complicated ones, but let's just keep track of this as I go. So it's a short piece of single-stranded DNA. should say this is what the probe is. So sorry, I'm putting that on the beginning. So the probe is a short piece of single-stranded DNA labelled either with, well, I've actually given you any examples in the notes. Fluorescent compounds, yeah, said what, but the thing is with that, you can actually use a variety of different colours of fluorescence to show up different sequences. So you can actually try and track several things at once. So you can label with fluorescent compounds, and then all you've got to do is shine UV light on it and it lights up. So fluorescent compounds or radioactivity. Now, if you have a look on the sheet, you'll see what's the most common radioactive isotope that they use. If you look down in the paragraph at the bottom, the probe's most commonly labelled with either radioactive atoms, the most common being, yeah, phosphorus 32, we would say. Phosphorus 32, well, I'll write it the other way rather than... Okay, phosphorus 32. Where would that be found in the molecule? Well, what sort of molecule are we looking at? Single-stranded DNA. All right, if you look at the model, whereabouts is the phosphate? Yes, in the backbone. You've got the sugar phosphate backbone. So I think on this particular model, it's the little purple bits. So if every little purple bit is labelled with radioactivity, just here, we're going to be able to find it. You know, so it's a neat little trick, and it's not terribly complicated, but it's the implications actually which are quite interesting. So you've got the probe will then bind to its target. And it's more about these bits of terminology, I think, than anything else. And that's known as hybridization, which word insists on spelling with a Z, of course. So it takes me a lot of time to track down the Zs and get rid of them. And if you look at the diagram that I've given you on that sheet, so you start with your chromosome, you've got your molecule of DNA separated out, and you get the DNA's been denatured, separated, you know, the hydrogen bonds have been broken, and then the probe, which has got little sort of semicircular lumps on it to indicate its labelling, will just bind to that particular bit. So the chromosome, you know, will then show fluorescence or radioactivity in that particular area. Now, in terms of how this can be used, we'll come on to that in a minute, but there's a lot of other processes or procedures that are sort of associated with this. And at the bottom of this, I've said, find out about. Now, I'm not going to ask questions in the exam on this, but if this is an area that interests you, or if it comes up on your presentation, then you will need to. But southern blot and northern blot are just two techniques that those I'm definitely not asking about. All right, so they are just to do with how the labelled chemical is transferred from sort of culture plates and that sort of thing. Now, the other one there, fish, this in itself is actually a potential topic, I would say. So I'm going to add it to the board. And that does not mean fish generally. All right, so fish as a topic in this context means fluorescence in situ hybridization. Now, fluorescence, we've met that. Hybridization, we now know what that means. The in situ bit actually refers to it being done in functioning cells, or, you know, within a cell in situ means, you know, on site. It's not just from extracted DNA samples. So FISH is about actually tagging the DNA inside a cell, in situ, rather than extracting it. So FISH as a technique is quite an interesting one. The other diagram sheet that you've got is, this is the transfer, and I've got a sneaking feeling this is the Southern Blot one, but I really ought to check. But you start with, you've got a plate with your tagged cells in. You press a filter paper against that, so you end up with the cells blotted onto the filter. You, oh no, they're not the tagged ones yet. You treat the filter to open the cells that are on there, denaturing the DNA, that's separating the two strands, breaking the hydrogen bonds. And you then end up with your single strand of DNA that will then stick to the filter. Then you add the DNA probe, I think, at that point. That shows up the gene of interest, and if it's been labelled radioactively, which this particular one has, then all you have to do is lay the filter onto photographic film, because radioactivity will obviously expose photographic film, you know, like an X-ray does, and that will then give you a pattern of where the radioactivity has been found, and you can then start, well, you make a comparison with your original plate so that you know which parts of the plate, and this is, it does say colonies, so that's going to be a bacterial plate. You'll know which of the bacteria contained a certain gene. So this would be a way, for example, of identifying genes in bacteria that might have given rise to antibiotic resistance. You know, although you'd be able to do that, you know, you'd know they were antibiotic resistant by growing them in the antibiotic, but actually locating the gene in which colonies would, you know, this would be a technique that you could use. Now, I'm not giving you as much detail on this as I could do, but there is a reason for that, which is, you know, that I'm pretty sure this will actually form part of some people's presentations. So I'm going to use, oh, it's another one of the McGraw-Hill ones, but it's only fairly short. Okay, let's just make this somewhat bigger.
At high temperatures, double-stranded DNA will denature or separate into single strands. When the temperature is lowered, the two strands will anneal because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also anneal. This is called hybridization, to indicate that each strand of DNA came from a different source. Regions of double-stranded DNA in which the respective single strands hybridize to one another are said to be homologous, or have similar or identical nucleotide sequences. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface such as nitrocellulose, homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various DNA probe techniques. Non-homologous DNA will not attach.
Fairly simplistic. So you have to bear in mind these are supposed to be higher education ones. I'm not convinced myself. But that's not telling you anything particular. I've got another one to show you, which is more about the metabolism of cancer cells, which is it's a little bit more applied, perhaps. But this one has underneath it, and that's the thing with these McGraw-Hill animations. Further down, they have these little quizzes. This is why you have the little whiteboards, all right, so that everybody can answer all of them. So you're just going to write a letter on them and hold them up, and we'll see. We'll see how we do. So I get everybody answering at once, don't I? You see. can always tell when we've got observations of any sort, can't you? All you've got to do is write the letter of the answer. So I'm not giving you all of them at once. Okay, first question on here. In hybridization, and then we've got 4 options. Option A is that 2 DNAs from the same source combine. Option B is that DNA from 2 separate sources combine. C is that DNA combines with complementary RNA. And D is that DNA is split into 2 separate pieces. All right, so hold up your answers. And you have to write big. Yeah. You were right, Chloe. Yes. It is, ah dear, it is B. So, yes, mostly right. Yeah, as he had the 2 separate sources combined. That's what hybrids are, from 2 separate sources. Yeah? You see, this is supposed to work. I just have to hope they play the game properly this afternoon, don't I? Right. Next question. DNA probes are used to A. Locate complementary RNA sequences in a test sample. B. Make homologous DNA. C. Make homologous RNA. D. Locate a specific DNA nucleotide sequence in a test sample. Yeah, I don't think that one was quite such a challenge. Come on, go for it, go for it. Taylor, come on, choose a letter. Choose a letter. D. It's fine. It's tracking down a particular gene. Yeah. Right. What? Did what? Oh, I don't know. That's right at the beginning of a year, wasn't it? You see, that's it's my very first thing that I do on those boards is what is life. Right. The next bit. Yes, if you think right back to September, that was the first bit of biology you did was what is life. Okay, next question. For DNA probes to hybridize. A. DNA strands must be homologous, having regions with similar or identical nucleotide sequences. B. DNA strands must be non-homologous. C. DNA must be complementary to RNA. And D. I don't know if I got those the right way around. DNA must be fragmented by reverse transcriptase. Remember they've got to match. So just final answer is it's A. A, the DNA strands must be homologous, so they've got to bind to each other with complementary base sequences. That's interesting, actually, isn't it? Because that's actually slightly misleading. I can see why you wouldn't have gone for it, because having regions with similar or identical nucleotide sequences, they're actually complementary, aren't they? Not the same. So that is a slightly confusing question, that one. And the complementary idea in here, the thing is, none of this involves RNA. So we know that's not the answer. But that's, I would say that's a dodgy question, that one. Okay, non-homologous DNA will attach to a DNA probe. False, yeah. At high temperature, 2 complementary DNA strands will anneal, join together. No, the high temperature did something. B, yes, B, the false one, because you needed the low temperature. So, yes, a high temperature separated them. So we should have that one. It doesn't want to scroll through. I've got to do it down the side. We should have that one. I mean, I'm hoping, you know, am I assuming that I'm right? I may be completely wrong with something. Non-homologous will attach, false, and at high temperature, that was false. So let's see. Oh, how exciting! Yes, we were right. You know, it's just one of those really sort of, you know, oh, well done. Yeah, sort of. You don't get any, you know, sort of fireworks or music or anything with this one. So. There is another little animation. This one doesn't require the whiteboards, but this I was looking for different ones, and this one is a completely different website, and I hadn't used this one before. So I've got to find the animations in there. And so we want that narrated. Okay. And it doesn't seem to want to play. Oh, that's because I hadn't clicked.
DNA chip technology
Right.
permits an investigator to rapidly determine which genes are expressed by a cell or tissue. To understand how a DNA chip works, let's consider an experiment in which we want to compare the genes that are expressed in normal and cancerous tissue. When a gene is expressed, it is transcribed into mRNA, shown here in green. In this experiment, the mRNAs from the two tissues are isolated and converted into complementary strands of DNA called cDNA by the enzyme reverse transcriptase. To distinguish between the two pools of cDNAs, they are fluorescently labeled with different colored markers. After the RNA is removed, investigators can use a DNA chip to compare the two cDNA samples. A DNA chip can contain an array of 60,000 or more different DNA sequences called probes. The probes are single-stranded and typically 20 nucleotides long. Each represents a tiny but unique region of a particular gene in the genome. The cDNA samples are mixed together and added to the chip. cDNAs that are complementary to the probes on the chip will bind or hybridize with the DNA, and thereby stick to that location on the chip. Unbound cDNAs are washed away. A scanner detects patterns of hybridization by sensing the fluorescent signals. In this example, a green spot indicates cDNAs from the cancerous tissue. A red spot indicates binding of cDNAs from normal tissue. A yellow spot indicates that cDNAs from both tissues bind to the DNA in that area of the chip. Because each area of the chip contains a known DNA sequence corresponding to a known gene, the identities of the hybridizing cDNAs can be determined. Using this data, investigators can establish which genes are expressed differently in the cancerous tissue, and thus may be able to design better treatment strategies.
All right, so that's an application. So, you know, if you're trying to make a comparison between normal cells and abnormal cells because of the way they express genes, then, you know, you can identify very specific genes. And very often, and cancer's a really good example, you know, it's not always going to be the same gene with the same mutation. So, you know, if you can identify the exact mutation, then you might be able to identify the exact protein that's then causing problems in cell division. And the more you know about the sort of molecular basis of somebody's disease, the better you can actually target the treatment. And that leads us really to sort of one of the main applications of using gene probes, and that's, first of all, you can screen people for genetic conditions with it, can't you, quite easily. You know, if you are looking, let's say, just in genetic counselling, you know, if you've got two parents who think they might both be carriers of something like cystic fibrosis, well, how would you find out? You know, how do you take DNA from each of those people and find the cystic fibrosis allele? Well, you do this, you know, and one might have it and one might not, or they might both have it, or neither might have it, but you can do it using this. And that allows you then to advise people on, you know, what their options are. Similarly, of course, embryo testing. that people can have IVF and select embryos that don't have certain genetic conditions. You can take one cell from an 8-cell embryo and test it. You know, you can analyze its DNA and find these genes. And again, you know, you'd use gene probes for this. And then you've also got, becoming more and more high profile, I suppose, in biological news type stuff, you've got this idea of personalized medicine, and particularly when it comes to treating cancer, because they're starting to look, aren't they, at the actual genetic nature of individual tumours and actually personalising medicines that will target that particular, perhaps, I don't know, an antigen on the surface or something like that of the cancer cells. So there's an awful lot of scope for that. But of course it opens the floodgates of arguments and ethical sort of objections, doesn't it, really? And it's something, you know, that you may well have your own opinions on. You might disagree with people on, but it's, you know, it's something that you've always got to consider. Where's that gone? Oh, it's up there.